2020, we're working on our microbial genetics. We just finished the two-part videos, DNA replication, and now we're going to take a look at how the information stored in the DNA base sequence can be used to guide protein synthesis. So we're going to describe this two-step process as gene expression. So we'll first talk about how DNA will act as a guide to make complementary RNA. And then in a second video that we'll call translation, we'll see how the base sequence in a special mRNA, a messenger RNA, will be used as a guide by the ribosomes by the cell to make protein. All right, so again, the first step in gene expression is going to be transcription. This is where DNA acts as a template to make the RNA. Okay, and we'll only be talking about three transcription products, three RNAs, M or messenger RNA, T or transfer RNA, and R or ribosomal RNA. We now know there's many more RNA products, but we'll just be talking about these three. And do remember, folks, in our introduction to the microbial genetics, we talked about the process called reverse transcription. For example, the retroviruses drive information flow backwards in cells. So the retroviruses, such as HIV, they're RNA viruses. They use their cool enzyme called reverse transcriptase to use their HIV RNA as a guide to make DNA, and then they slip the DNA into our chromosomes. But again, folks, we'll come back to this in the viral unit. We're talking about cellular transcription right now. So this is our overview of transcription. So first of all, the enzyme that performs transcription is called RNA polymerase. And in cells, RNA polymerase use single-strand DNA as a template to make complementary RNA. The transcription will begin when the RNA polymerase binds a special DNA sequence called the promoter. So in some ways, we could say the promoter is where transcription will begin. Transcription will proceed in a 5' to 3' direction. RNA is synthesized 5' to 3' just like DNA is, and the RNA is synthesized anti-parallel to the template strand, so a lot of similarities to DNA polymerase. And then transcription will stop when the RNA polymerase hits a special DNA sequence called the terminator sequence. So again, we're going to focus on E. coli. We'll be focused on bacterial transcription, bacterial RNA polymerases. And bacterial RNA polymerases are really cool. They're made of two subunits. And the one of the subunits is called the sigma subunit or sigma factor. This is like the brains of RNA polymerase. And then the second subunit is called the core enzyme or core subunit. This is the actual workhorse of RNA polymerase. This is the part that actually synthesizes the RNA. The reason we say that the sigma factor is the brains, is that it's the part of the RNA polymerase that can recognize that special DNA sequence called the promoter. So it's a sigma subunit, the sigma factor, that gets RNA polymerase to bind at the correct site on the DNA called the promoter, so that transcription can be initiated. And initiate is just a fancy word for to begin or to get started. And then once RNA polymerase has synthesized a short strand of RNA, we'll just say maybe 10 to 12 nucleotides, the core subunit now knows where it's supposed to work, right? So sigma will fall off, right? And then the core subunit continues to carry out transcription until it hits that termination of transcription signal, and then it will fall off. It will release the DNA, it will release the RNA, and transcription's over. This is just, to me, kind of cool. So in bacteria, they'll have different types of sigma factors, sigma subunits. It will recognize different promoters on the chromosome. And this is one way that the bacteria can control which genes will be transcribed and eventually translated. So that's just kind of a neat control process. We're always interested in unique microbial targets for our magic bullets, right? Our antibiotics. So bacterial RNA polymerase is one such unique microbial target. So a class of antibiotics called the rifamycins, and a specific rifamycin is called rifampin. These are similar to macrolides in structure, and we'll see that macrolides actually have another target. They're going to knock out the bacterial 70S ribosomes. So the rifampin target is different. It's going to inhibit the bacterial RNA polymerase, which will block transcription and thus will block protein synthesis. Rifampin is really important as a frontline drug against mycobacteria, so Mycobacterium tuberculosis, Mycobacterium leprae. So patients that are suffering with tuberculosis or Hansen's disease, rifampin is often a frontline drug. One of its advantages is it has really good penetrating properties. It can penetrate tissues. It can reach therapeutic levels in cerebral spinal fluid, which is often hard to do. And this is really important. It can reach therapeutic level inside of abscesses. Often abscesses have no blood supply, hard to get therapeutic levels of antibiotics within the abscesses. And furthermore, and this is really important when you're treating mycobacterium infections, we're going to see that the mycobacteria can actually replicate inside our macrophages. So it's really important that we can get high levels of rifampin inside the macrophages. And indeed we can. We can get therapeutic levels of rifampin in the macrophages to inhibit the mycobacterium. One thing though you'd want to warn your patient and their family and friends is that the rifampin itself is this deep, deep, beautiful red color. So when you're giving it to your patient, they're going to have this orange-red feces, urine, saliva, sweat, and tears. And sometimes they might panic and think that they're bleeding, but it's not. It's just the pigment of the rifampin that's coloring their urine, saliva, feces, sweat, and tears red. So just as we had DNA replication rules, we have bacterial transcription rules, and a lot of this will be repeat. So we want to remember that within cells, the bacterial RNA polymerase is going to require a template, and that will be single-strand DNA. At any specific site on the chromosome, we want to remember the chromosome has 2 strands of DNA. Usually only one of the strands will be transcribed. Now, again, viruses love to break all our human rules. So some viruses will violate this rule, but we'll say in general, in cells, in bacteria, usually just 1 strand of DNA will be transcribed at any one gene location. So the strand that's transcribed, the DNA strand that acts as a template is called the template strand, and the opposite strand is called the non-template strand. RNA is synthesized 5' to 3' anti-parallel to the DNA template. We'll need those energized nucleoside triphosphates as the building blocks. And again, folks, we're just going to talk about these 3 transcription products: messenger RNA, transfer RNA, and ribosomal RNA. And there's many other functional RNAs, but we're just limited to these 3. This is detail you need not remember, folks, but the promoters, those are the sites you'll remember where sigma factor binds to start transcription. They tend to be conserved, meaning they have promoters on different genes, maybe sometimes promoters in different bacteria. Their DNA sequence is often really similar. They talk about the -10 upstream from where the transcription actually starts. It's sometimes referred to as the Pribnow box or Tata box. I would never ask this on the exam. And then there's another conserved region, 35 nucleotides upstream, where transcription will begin. And again, just a conserved DNA sequence. So we'll say, and this isn't exactly right, but we'll go with it. We'll say promoter is a DNA sequence that will help start transcription, and then the terminator sequences are the DNA sequence that will stop transcription. So in class, we'll be able to use the whiteboard, and what I like to do is show transcription only showing the template strand. So sometimes the second DNA strand makes this a little bit confusing, but we can figure out, you guys, right here, if this is RNA polymerase, that's the core subunit, there's sigma factor, here's our double-stranded DNA. We can figure out that this top strand is what's going to be the template strand, because remember, the RNA polymerase has to make the RNA anti-parallel to the template in 5' to 3'. So just by orientation, I know it's this top DNA strand is a template strand. So when RNA polymerase binds, it is really cool. It can act as its own helicase, meaning we don't have to have a separate helicase. In here, RNA polymerase can break the hydrogen bonds between the 2 parent DNA strands, and then it will transcribe just one of the strands. So again, here's our bacterial RNA polymerase, the core subunit, and then the sigma factor, the brains. And again, the sigma factor is going to recognize that promoter and is going to get the core subunit to start transcription after they bind to the promoter. Here's the terminator sequence. So here we see the RNA polymerase acting as its own helicase. It breaks the hydrogen bonds, creating the single-strand DNA template. So that's going to be this one up here. Okay, so it's unzipping the double-stranded DNA. Okay, so this is the initiation of transcription. Initiation just means the beginning. This is just in more detail, folks. So here's the template strand, and we can see that this is our growing strand of RNA, complementary RNA. So the RNA polymerase is going to introduce the correct nucleotides carrying the complementary bases here. And do remember again that the RNA polymerases, they don't need a primer and they don't proofread, so they can make quite a few mistakes, but the cells usually can survive that. And do notice that here's the RNA polymerase here, and we can see that it's broken the hydrogen bonds here. As soon as it moves on, the DNA reforms the hydrogen bonds, right? So the RNA polymerase unzips the DNA, and as soon as it moves past, then the DNA, it gets zipped back together again. Those complementary bases will form their hydrogen bonds again. And here's just showing the RNA polymerase. It's hard to see here hitting the terminator DNA sequence here. And when RNA polymerase hits the terminator sequence, it releases the DNA. Oops, where's my? It releases the DNA, and it also releases the RNA transcript right here. Okay, so much easier than DNA replication. So this is termination when it stops. And this is just one more time, folks, just to show you how efficient the bacteria can be. Once an RNA polymerase has moved off the promoter and starts transcribing, another RNA polymerase can bind. So you could have several RNA polymerases transcribing the same DNA gene here. Here's the sigma factor, and we can see after a little bit of RNA is synthesized, the sigma factor, its job is done, it falls off, and it will go join another core subunit. The sigma factor just made sure that the RNA polymerase core subunit started transcribing in the right place. So this could be, for example, you guys, our messenger RNA that's being made here. And then as soon as we hit the terminator, again, the RNA polymerase would release the DNA and release the RNA transcript here. And again, folks, we're just showing that the RNA is being made 5' to 3', and it's anti-parallel to its template. So again, this would be the DNA template strand. This opposite strand, the non-template strand, sometimes they refer to it as the coding strand. But for us, you guys just remember one of the DNA strands will act as the template. Now, polycistronic messenger RNA, this is unique to prokaryotes. Eukaryotes like ourselves don't make it. And if I can hopefully describe this here, you guys, this would be the bacterial chromosome. Notice there's a promoter there. There is a gene, say for one protein here, and then notice there's no transcription terminator signal, and then right next to it is a gene for another protein, no transcription terminator, and then a gene for a third protein, and it's only then that we hit a transcription terminator. So what this means is, in bacteria, if RNA polymerase binds here, it's going to make one long RNA transcript that will contain information for gene 1, gene 2, and gene 3, or for protein 1, protein 2, and protein 3. So here's our messenger RNA, right? So here's the mRNA sequence that when it's translated by the ribosomes will have coordinated synthesis of these three genes, these three proteins. So we're going to have protein 1 made, protein 2 made, protein 3 made before oh, excuse me. Gosh, you guys, I'm so sorry. Let me back up here. Let's look at this polycistronic messenger RNA, meaning it carries information for 2 or more genes. So this is kind of jumping ahead to translation, but this first part of the messenger RNA, the 5' end, there'll be what's called a ribosomal binding site, and that's where the ribosome will bind. And then as soon as it recognizes the first, this is called the start codon AUG, the ribosome starts translating the messenger RNA into the amino acid sequence of a protein. And then when the ribosome hits, this is called a stop codon, it falls off. and then that releases this one protein, right? Then there'll be another ribosomal binding site. The ribosome will bind, and then it's going to translate this second um, uh, mRNA that encodes the second protein, right? The ribosome hits the stop codon, it falls off, right? And that releases the second protein. And then we'll have another ribosomal binding site right here. The ribosome binds, and it will translate the third piece of mRNA here, and it hits the stop codon, and then we um, release our third protein. Now, this polycistronic messenger RNA carrying information for more than one gene product, for example, in this case, three proteins. This is really cool. This is a way that bacteria can coordinate protein synthesis. Say proteins, maybe all three of these proteins are involved in the same metabolic pathway. Or maybe one of these proteins is a transport protein for a substance that will be transported into the cell, and then the second protein is some catabolic enzyme that will hydrolyze whatever the first transport protein has brought into the cell. So later on, when we talk about bacterial operons and control of operons, we'll see that very often we're going to be talking about this polycistronic messenger RNA encoding information for two or more products, such as two or more proteins. Okay, but again, this is going to occur only in prokaryotes, not eukaryotes. And one reason why the polycistronic messenger RNA might not occur in eukaryotes is that transcription is so much more complicated in eukaryotes, such as yourselves. So we want to remember, folks, that in a bacterium, where is the chromosome? It's in the cytoplasm. Where are the ribosomes in a bacterium? The ribosomes are in the cytoplasm. So we'll have transcription and translation in bacteria occurring both in the cytoplasm. But in contrast, you guys, we want to remember in eukaryotes have a membrane-bound nucleus where the chromosomes are. So transcription in eukaryotes will occur in the nucleus, right? And then eventually the processed RNA, it takes us a while to make our messenger RNA. The messenger RNA will then be delivered into the cytoplasm where our cytoplasmic ribosomes are located, where translation will occur. So in eukaryotes, transcription occurs in the nucleus and translation occurs in the cytoplasm. So transcription and translation are separated in time and space. Therefore, as eukaryotes, we aren't as efficient at protein synthesis as our little bacterial distant cousins. And then, folks, as we can see here, transcription, again, in eukaryotes like ourselves, it's so much more complicated. If we look at our chromosomes, we'll see that there's alternating DNA that represents exons and introns. And so when this piece of DNA is transcribed by eukaryotic RNA polymerases, we'll just call this an RNA transcript. This isn't messenger RNA yet. And what we'll see is the RNA that was transcribed from these introns. So we could call these little pieces of RNA intron RNA. So there's intron RNA there, intron RNA there. Within the nucleus, the intron RNA is going to be cut out, and then the ends of the exon RNA are going to be spliced together. So this is some major editing going on in eukaryotes that we do not see in prokaryotes. And then furthermore, once we have our spliced mRNA, the cell further chemically modifies both ends. And it's only after this cutting and splicing and chemical modification that we have mature messenger RNA. And it's that mature mRNA that will travel into the cytoplasm where the eukaryotic ADS ribosomes will bind and then start translating the messenger RNA. So again, folks, in eukaryotes, transcription is so much more complicated, and it might be because of all this cutting and splicing and chemical modification that eukaryotes can only make monocistronic messenger RNA, and monocistronic messenger RNA contains information for only one gene product, for example, only one protein. I think that might be it. Right, okay, so folks, we'll stop here. So this will be our transcription video, and then we'll tackle translation. And this is a much more complicated process. This is where our messenger RNA is going to act as a guide, a template for protein synthesis. So we're changing languages here, and you can imagine it's really complicated. We're going to tackle it like an onion, a multi-layered onion. We're going to start with the outer layer, just look at the basic accomplishments without looking at details. And then once we understand the kind of the most important steps in translation, then we'll start peeling the layers of our onion back, and we'll look at more and more details. Okay, just to get us warmed up here, translation, a fancy term for protein synthesis, will occur where in the cells? It's going to occur at the ribosomes. And again, folks, we're going to be using our good old E. coli is our model organism to explore bacterial translation in the next video.